CRISPR is a microbial antiviral defense system. The Cas9 CRISPR protein has been widely applied as a genome editing tool. In this session, the basics of CRISPR as an antivirus mechanism are explained. It's shown how CRISPR can adjust to new viral infections and adequately combat viruses. In the last part of this session, we discuss how autoimmunity can be avoided. Viruses are the most abundant entities on this planet and outnumber their host at least by a factor of 10. Consequently, in their natural environment, microbes are under constant danger of virus infection. To defend themselves against the positive-negative impact of viral infection, bacteria and archaea, like humans, have evolved a range of different defense strategies. This slide summarizes the known viral defense strategies. The first to block adsorption, the second abortive infection, the third is to block uptake, the fourth is the restriction modification system, and the fifth is the CRISPR system. The CRISPR system is present in about half of all bacteria and most of the archaea. There exist many different CRISPR systems, CRISPR types, of which the Cas9 based system is the most famous. The basic mechanism of defense is largely similar for all families. The differences rely on the different CRISPR-associated or Cas genes used to classify the several families. Some systems target foreign DNA, whereas others target RNA. The discovery of CRISPR started with the first description of clustered, regularly interspaced palindromic repeats in the genomes of bacteria and archaea. For long, these repeats were seen as uninteresting genomic regions until in 2005, several groups simultaneously reported that the 30 base pair spacers in between these palindromic repeats contained sequences that were matching exactly to those of viral genomes. At that moment, it was proposed that this CRISPR array might be part of an antiviral defense system. This slide depicts a typical CRISPR array. CRISPR arrays can contain between 2 and several hundred of repeats. Depending on the CRISPR family, the size of these spacers and repeats varies a little. Several different CRISPR arrays can be present in a single bacterial or archaeal genome. Each spacer is unique and matches to an individual virus or extrachromosomal element like a plasmid. The Cas genes are often encoded in close proximity of the CRISPR arrays. First proof that CRISPR indeed functions as an antiviral defense system came from research on phage-resistant bacterial strains in the dairy industry. Researchers showed that the only difference between a virus-susceptible and virus-resistant strain was the presence of one additional spacer in the CRISPR array of the resistant bacteria, shown in red on this slide. This spacer matched 100% to a region in the viral genome to which a strain had gained resistance. This region is called the protospacer. The role of the PAM will be explained on the last slide. The addition of new spacers matching a certain virus will render the strain resistant in future. As these adaptations in the CRISPR system are made in the genome, this resistance will also be inherited by the offspring of the strain. As such, the CRISPR system functions as an adaptive and an inheritable defense system. In this slide, the first phase of the CRISPR defense mechanism is demonstrated, with the example of Cas9. This first stage of CRISPR-mediated defense is defined as the adaptation phase. After infection with a virus, the foreign DNA is recognized and cleaved by the Cas proteins. A small portion of the genome is incorporated as a new spacer in the CRISPR array. New spacers are generally added in the CRISPR array near the leader sequence, which is shown in gray on this slide. As such, the CRISPR array gives an overview of past viral encounters of a strain. Spacers are occasionally removed from the CRISPR array by recombination events that can occur in the middle or towards the end of the array. Second phase of the CRISPR defense mechanism, the CRISPR array is transcribed in a long RNA molecule. Cas9 can process the pre-CRISPR RNA, and in other types of CRISPR families, this RNA processing occurs by different Cas proteins. Cleavage of the pre-CRISPR RNA occurs with the help of a trans-activating CRISPR RNA or tracer RNA. This tracer RNA hybridizes specifically with the repeats in the pre-CRISPR RNA bound by Cas9. This event recruits a host encoded RNase that eventually processes the pre-CRISPR RNA in small guide sequences. In other types of CRISPR systems, this cleavage can also occur by a Cas protein. The guide RNA sequence remains bound to the Cas protein. In the third phase of the CRISPR defense mechanism, the virus genome is targeted and eliminated. The Cas9 guide RNA complex scans cellular DNA using its CRISPR RNA containing a single spacer as a guide. As soon as it finds a perfect match, the DNA is being cleaved, and as such, the viral infection is halted. The 30 base pair sequences provide enough specificity to not target the bacterial or archaeal genome. However, in theory, there is one position with a perfect CRISPR RNA match in the bacterial genome. This is the CRISPR array itself. Targeting of this region could lead to autoimmunity. To avoid autoimmunity, the PAM sequence is important. On this slide, the mechanism of PAM is explained. PAM stands for protospacer adjacent motif. Usually it consists of only a few base pairs. It's located adjacent to the protospacer in the viral genome, which is shown in red. As a rule, it's not present adjacent to the spacer in the CRISPR array. DNA cleavage by the CRISPR RNA-loaded Cas complex only occurs when a PAM is present. Therefore, the PAM represents an effective mechanism to avoid autoimmunity.